Hey guys, we're going to take notes on cell transport and osmosis. So this would be transport of substances into and out of a cell across the cell membrane. There's a lot of good videos, short ones, to show the movement for each of these types of transport. I'll share some of those with you, but you can also go to YouTube and find a whole bunch, and I would recommend doing that and looking at the video so you actually see the motion of how these substances get across, so you can compare one type of transport to another. So let's get going here. So passive transport. This type of transport across the membrane does not require any energy, so no energy is needed. Active transport requires energy, and the type of energy that your body uses is a fuel called ATP, which is short for adenosine triphosphate. So we'll start with passive transport. There are different types of passive transport. The first one is simple diffusion. Now, because passive transport does not require any energy, why is that? Well, it's because these substances move from a high concentration to where there's a low concentration. And think of it like the natural flow, like compare it to like a river, right? So a river is flowing in one direction, and it's kind of moving from high concentration to low concentration, or think of it as going downstream. So as that river flow, it doesn't require any energy. If you were swimming in that river and you were floating, you would flow downriver. Doesn't require any energy for you to move. All right. So that's kind of hopefully will help you with remembering that. So simple diffusion is diffusion of very small molecules. They can be either polar or nonpolar. The little picture here on the right, this is representing the inside and outside of the cell. So, maybe on the left side, we can say that this is the outside, and on the right side is the inside. Okay. And the dotted line represents that cell membrane, that semipermeable membrane. So you have these blue dots and the red dots. And the blue dots you see are kind of big. These are big molecules. They can't get across that cell membrane. They're too big. But the red dots can. And by definition of passive transport, if a substance moves from high to low concentration, those red little molecules, whatever they are, let's say they're sugar, okay, sugar molecules, they are going to naturally want to move across the membrane from that left side to the right, because as a rule, by definition, they're going from where there's a high concentration of them to where there's a low concentration. And they will continue to move back and forth until it is equal on both sides of that membrane. Facilitated diffusion is the next type of passive transport. And this is a little different because in this diffusion, it's through a protein channel. So remember when we looked at the structure of a cell membrane, there's these different proteins, these transmembrane proteins, and they have a channel or like a gateway. And substances move through that little channel or tunnel, and that's how they get in and out of the cell. Because it's passive transport, these substances will move again from a high to low concentration. In this graphic here, these are both different types of protein. One is a protein channel, the other one is a carrier protein. They work a little differently, but they're both an example of facilitated diffusion. So the little red dots on the left, right, there's a higher concentration of them on that side of the membrane and a lower concentration on the other side. So those red molecules, let's say it's sugar again, are going to want to move to where there's a low concentration, and they're going to move through that channel. That's how they get across. The carrier protein kind of works same way, more or less, but it's sort of like a gateway that opens and closes. Okay, so passive transport moves with the gradient. It moves with the natural flow. Okay, pause if you need to. I'm going to move on otherwise to active transport. Active transport requires ATP. Why does this require fuel or ATP? Well, because it's moving against the concentration gradient. It's moving in the opposite direction of the natural flow. So imagine you trying to swim upriver or against the natural flow of the river. It's going to require you to use energy, right, to be able to swim in the opposite direction, just like it would require a lot more energy for you to try to run up a hill than run down a hill. So we're going from low to high, as you see in this picture. And because we're going against, that's where the ATP comes in. And that fuel is needed to get those molecules across. Where is this used in the body? The sodium-potassium pump is what we call a membrane pump. Sodium and potassium are things that you get in your diet and are very important for your body to work. And so certain levels are needed. For example, they're important for your heart to beat. So these things do need to move across a membrane, and ATP is the fuel that helps get those to where they need to be. Now I'm going to go back to passive transport. There is another type of passive transport called osmosis. This is actually the diffusion of water. So you can apply the same definition to osmosis as we said in diffusion above. No energy is needed. What is different about osmosis is that this is specifically only talking about water moving in and out of a cell. Again, it's going from a high concentration to low concentration. No energy is needed. Let's look at this graphic. This is one that is commonly used. So like if you were going to Google video or animations of osmosis, they oftentimes use this little YouTube looking thing to show the water levels on two sides. Again, here in the middle is what is representing the cell membrane. And so you have things on the left side of the membrane, which could be inside the cell, let's say, and on the other side of the membrane, which could be outside of the cell. The blue is representing water. And again, the little red dots, we're just going to say that they're sugar molecules. Okay, also referred to as the solute. So the substance that is in the water is the solute. The water would be considered the solvent. Okay. So if I asked you, where is there a higher concentration of sugar? You would count up those dots. And obviously, if we counted them up, we would see that on the right side, you have a lot more sugar, higher sugar than on the left. Well, what does that say about the water concentration? Well, if you have a lot more sugar on the right, then that means you have less water on the right. And by the same token, on the left, if you have a lower amount of sugar or a solute, that means there's actually more water on that left side. Okay, so then the question here is, if we're talking about osmosis, and I asked you, well, which way is the water going to move? Is it going to go from left to the right or right to the left? So think about it. Look at the definition. Where is there a higher concentration of water? And then which way will the water move across the membrane? All right, if you said that the higher water is on the left, you'd be correct, and the water would move to the right. All right, so it's going to move from the left to the right until they are even. Now, what's going to happen here, and you can't see it in this picture, if I gave you a after picture, the water level would actually rise up. So the blue would actually go up once this was done. That doesn't mean that there's more water on the right side. It means that it would equal out, so the actual concentration of water would be equal on both sides. You can kind of think of it as the water moving to the right to try to dilute all that sugar that's there. All right, that brings us to talking about tonicity. Tonicity is solute concentration. In other words, how concentrated is the solute that you're talking about? 3 descriptive words to describe tonicity: hypertonic, hypotonic, and isotonic. All right, and this is kind of a tough one. Students get these confused. It's very easy to get confused. So it's just something you have to kind of practice looking at and then memorizing. When you're looking at something that is hypertonic, that means there's more solute and therefore less water. Hypotonic, less solute, more water. And isotonic would be equal solute concentration. So let's see what this would look like if we looked at an actual cell. Here we have, we're going to look at an animal cell, and then here an example of a plant cell. They're very similar as far as what happens. So here on the left we have, and let's look at the animal cell. That happens to be a red blood cell. We're going to look at this red blood cell, which is in a hypotonic solution. Okay, hypotonic solution. So that means there is more water and less solute. So if there is more water outside of the cell, which way is that water going to move? Well, the arrow here shows you the water is going to move in to try to dilute that solute that is inside. What is going to cause, what's going to happen is by that water moving, it's going to plump up. You see that cell kind of gets big, right? Almost like you blew it up with air. And actually what could happen is that the cell could burst in certain situations. Now in the isotonic, here, this is what a normal kind of a shape of a red blood cell would be. And you have equal amounts on both sides. Now, if you put a red blood cell in a hypertonic solution, which means it has less water here on the outside in the blue and more solute, that cell is then, the water in that cell is going to want to move out. It's going to leave the cell to try to dilute all that solute outside of it. And as a result, it's going to shrivel up and shrink. All right, similarly, same thing happens in a plant cell. Plant cells actually prefer a hypotonic solution. And they won't burst because remember, a plant cell has a cell wall, has that extra rigid and layer on the outside of the cell membrane. And so this is what gives the plant cell its shape. So it's plumped up. You guys know what it looks like when you forget to water a plant for, let's say, a couple of weeks, right? It's going to shrivel up. That would be like a hypertonic solution example. But when you water it, those cells plump up and you have a nice kind of healthy plant. So a couple practice problems. This is something that you'll probably see on a quiz and a test. So here's our little membrane example with our U-shaped thingy. So on this side here, you have less solute, more water. On the right side is more solute, less water. So there aren't any dots drawn in here, but what you have is these measurements of solutes there. You have a certain amount of glucose and a certain amount of NaCl, sodium chloride. Add that together. This is how much is present on the left, and you have 1.2 on the right. Okay, so which way is the water going to move? Well, it's going to move from side A to side B. It's going from a low concentration, from a high concentration to a low concentration. All right, and here's a few more examples, again, where you would be seeing something like this on a quiz or a test, where you're shown how much, in this case salt, they're using the example, how much percentage of salt is inside the cell and how much is outside of the cell in the beaker, and then which way is the water going to move. Is it going to move out of the cell and therefore the cell is going to shrink up, or is it going to move into the cell and cause that cell to plump up? Okay, and then down here, I kind of give you the answer to that. So if you are describing the tonicity, this is how it would be described. And if I asked you to describe the cell itself, okay, and this is how you would describe that.